So I'm interested in the basic biology of aging and questions of, what are the determinants that influence how different people age? And I've been interested in this question since graduate school. And one of the things that I always find interesting about aging is the fact that we don't all seem to age exactly the same way. And at least by appearances, at least, so there's a couple of people who know who these folks are. I always wonder when I show this slide how many people are going to recognize these individuals. Yeah, that's right. I certainly, among the graduate students when I give this talk now, most of them don't know who Tina Turner or Keith Richards are until I point out, so many of us know Keith from the Rolling Stones, right? But then when I point out that he was also in the Pirates of the Caribbean movies, then they get it. So one of the reasons why I like this particular comparison, though, is it clearly makes the point that you wouldn't necessarily guess by looking at these people that Tina Turner is actually older than Keith Richards. It also makes the point that aging, like any complex phenotype, is a combination of genetic and environmental factors. And so I think Keith Richards is probably a nice example of this, that some of his environmental choices may have influenced his apparent rate of aging. And so, the research in my lab and many others in the field that I work in is really to try to understand what are these genetic and environmental components at a molecular level, and then hopefully use that information to intervene in the aging process at a molecular level. So that's kind of the big picture of what I work on and where my interests lie. And you can think about why this might be important and impactful if you think about disease. So if you think about the diseases that are the major killers in developed countries and also in lesser developed countries, these diseases are becoming more important as people are starting to live longer. If you think about these diseases, you can think about risk factors, right? And so we sort of know what some of the risk factors are for these diseases. But what I think a lot of us maybe recognize but don't really think about is that there is, in fact, a single greatest risk factor for all of these diseases. And it's not how much you eat. It's not how much you drink. It's not whether you exercise. It's not whether you smoke. It's how old you are. And it's by an order of magnitude, the impact of aging on your risk of these diseases relative to sort of these more modifiable risk factors. And I don't say this because I'm trying to argue that we should go out and drink as much as we want and smoke and stop exercising, because those are things that you can control. But I think it is illustrative to think about the fact that aging is really a permissive state for all of these diseases. And why is that? What is it about aging, again, at a molecular level, that causes this? And then if we are able to intervene in the aging process at some point, then it offers the potential that by modifying aging, we can, in fact, modify the risk of all of these diseases and disorders of aging, maybe simultaneously. So I realize that to some extent this sounds like science fiction, and what I'm going to tell you today is we're not quite there in terms of being able to do this in people. But in laboratory animals, it's actually now pretty routine to modify the aging process. And in fact, what we see when we do that is a delay in the onset and progression of multiple age-related phenotypes. And in more complicated animals, like mice and rats, who age in some ways much like us, we see a delay in the onset and progression of age-related diseases. And so the sort of best documented or characterized environmental approach to delaying aging is dietary restriction or caloric restriction or calorie restriction. And I sort of use those terms interchangeably. There are some nuances to the term dietary restriction versus calorie restriction. I'm happy to talk about that if anybody's interested. But at least for now, I'm just going to kind of use those terms interchangeably. And what we really mean when we talk about dietary restriction is a reduction in nutrient availability in the absence of malnutrition. And so obviously that latter part of that is important. But what's been observed is that under conditions of dietary restriction, aging seems to be slowed, longevity is increased, and this has been seen across a broad evolutionary distance in laboratory systems. And so 4 of those are shown here. And I'm not going to, this is going to be a data light talk, but I am going to show a couple of these survival curves where we've got the percent alive in a cohort now as a function of age. And if you look across these model organisms, what differs is the metric of aging. So the length of time that it takes for an organism to go from birth to death is different across all of these species. So in mice, we're talking months or years, whereas in fruit flies and C. elegans, we're talking weeks to go through the entire life cycle. And so that's one of the reasons why these simpler organisms have been very powerful in sort of identifying approaches to slow aging that can then be tested in more complicated and long-lived animals like mice. So, so in all of these cases, what you can see is that relative to the control cohort, both median and maximum lifespan is extended by dietary restriction. And in again, more complicated animals where they show signs of aging, it really seems to be the case that not only is lifespan extended, but health span, or the period of life spent in relatively good health, is also extended. And that can really be seen in non-human primates like rhesus monkeys, where dietary restriction has also been shown to extend both lifespan and health span. So one point I'll make, this paper is a few years old. I think this was initially published in Science in 2009. The same group, Rick Weindrich's group, has published a follow-up paper last year where the survival curves are out a little bit further. But even back then, you can already see that in terms of lifespan, the restricted group in red is living longer than the ad libitum fed group in blue. I'll point out this is an extremely long experiment. So we're out 30 years, and the control population is just getting to median survival. So this doesn't prove that dietary restriction is going to work the same way in people. But certainly this is much closer to humans than mice. I think we can all agree. And I think that one of the most powerful parts of this particular paper is that not only did they look at lifespan, but they looked at percent of animals that are free from age-related disease. And in this case, they're sort of lumping together cancer and metabolic disease and cardiovascular disease and saying if an animal shows one of those diseases, then we're going to say it has an age-related disease. And so what you can see here is that in the ad libitum fed group, about 80% of the animals have shown at least one age-related disease, whereas more than half of the restricted animals don't show an age-related disease. So again, at least consistent with the idea that aging has been slowed and the animals are healthier and living longer. Okay, so that's dietary restriction. We have lots of genetic ways to do this in laboratory animals, but if you're actually thinking translationally about the potential, at least, to in the relatively near future start thinking about translation to humans, what you really want is a pharmacological approach to delay aging. And so certainly the best case right now can be made for a drug called Rapamycin or sirolimus. I'm sure many of you are familiar with this drug. So this is a drug that was first identified from soil samples on Easter Island. And if you're interested, there's sort of a popular press article that was written in Bloomberg Business News recently that kind of lays out some of the history of rapamycin and its role in first as an anti-cancer drug, and as probably many of you know, it's used primarily clinically as an organ transplant rejection drug, and how that's now sort of morphed into a lot of interest in rapamycin as a drug that can impact the aging process. So rapamycin is FDA approved, mostly for transplant rejection, although it's also used for some rare cancers and in cardiac stents. And it first became of interest in the aging field, I would say back in the early 2000s, where my lab and others found that if we treat invertebrate model organisms with rapamycin, or if we genetically modify the mTOR pathway, which is the target of rapamycin, that we can extend lifespan in invertebrate species. But I think people really started paying attention when it was found that you could actually give this drug to mice and extend their lifespan. And I think most interestingly is this was actually done late in life, and you still can see an increase in lifespan. So this is a study, this is from a study done by the Interventions Testing Program at the National Institute on Aging, where they started giving mice this drug at 600 days of age. So that's about the mouse equivalent of a 60-year-old person. And when this experiment was first done, I don't think anybody thought it was going to work, because at that point we hadn't really identified any intervention that you could start in midlife or late life and still significantly increase lifespan in a population of complex animals. But it did work, and so what you can see here is that even starting at 20 months of age, you get a significant increase in both median and maximum lifespan. And I'll show you a little bit of data in just a second, very preliminary unpublished data from my lab that suggests that even as late as 28 months, and that's a point where about 75% of the population, the control population, has already died. If we start the treatment at that point, we can still get quite robust improvements in survival and health span. So that says that we can start giving mice this drug in midlife and get an increase in lifespan. Importantly, now lots of other follow-up studies have suggested that you can also get improvements in a variety of age-related diseases in mice by treatment with rapamycin. So in mouse models of a variety of cancers, mouse models of Alzheimer's disease, we can debate the merits of whether those are really Alzheimer's disease models, but certainly in 2 different models of Alzheimer's disease, rapamycin has been shown to improve outcome, obesity, age-related declines in cognitive function in mice, cardiac function, immune function. So a variety of age-related phenotypes seem to be impacted positively by treating mice with this drug, in addition to increasing lifespan. And a recent study from Novartis that I'll mention again in a minute has at least hinted that this might work the same way in people. So in that particular study, what they did was they took healthy elderly humans and they gave them a 6-week treatment with everolimus, which is Novartis's version of rapamycin. And the question they were asking is, what is the impact on decline in immune function with age? So as we all know, the response to an influenza vaccine goes down as people get older. So what Novartis did was they took healthy elderly people, gave them 6 weeks of everolimus or 6 weeks of placebo, and then gave them a flu vaccine. And what they saw was that the people who'd been taking everolimus had a better vaccine response than the placebo group. And this parallels exactly what had been seen previously in mice, and I'll show you the data in a minute, showing that rapamycin can improve immune function in elderly mice. Now we have some indication that that's probably true in people as well. So it doesn't prove that rapamycin is going to have this effect in people, where we can start giving it to 60-year-olds and we're going to increase their lifespan. But it's a hint that at least in some ways, rapamycin is working the same way in people as it is in mice. And the other point I'll just make is, if you're thinking about a translational intervention to impact healthy aging, what you really want is something like this, that you can start giving to people in middle age rather than something that you have to start treating people with when they're teenagers. First of all, teenagers don't care about aging at that point. But I think the risk of side effects, which is something we always need to think about, but particularly when we're talking about an intervention to treat otherwise healthy individuals to target something like aging, the risk of side effects is going to be much reduced if it's an intervention that people can start when they're in middle age, and possibly start in a transient sense. So maybe 3 months or 6 months of rapamycin would be as effective as continuous treatment. So these are the kinds of issues that the people in my field, including me, who are starting to think translationally about targeting aging, are keeping in mind as we move forward. So this is the study that I was just referring to in mice. This is work from Pan Zhang's lab at Michigan, where they were asking what is the effect of rapamycin on age-related immune decline. And so I'll just take you through this figure quickly. So this is, again, using an influenza vaccine followed by an influenza challenge as a measure of age-related immune function. So in this case, these mice are, I think they were 22 to 24 months old. They got 6 weeks of rapamycin or placebo. And then they were taken off the drug and they were immunized with a flu vaccine, and then 2 weeks later challenged with a live flu. Okay, so the old mice that got the vehicle treatment and then the vaccine and then the challenge are right here. And you can see that about 80% of them died following the challenge, as compared to young mice who got the vaccine. All of them survived the challenge. Okay, so immune function declines with age, and the readout here is that after a vaccine, old mice, about 80% of them die when you challenge them with flu, whereas young mice responded just fine to the vaccine and they all lived. And you can't really see it here, but it's kind of buried under the young mouse line. The old mice that had gotten the rapamycin treatment then were immunized and then challenged with the live virus all survived. So again, suggesting that somehow this transient treatment with rapamycin had rejuvenated the immune system so that, at least in this assay, it functioned like a young immune system. And that parallels very much what the Novartis study that came out about 4 months ago showed. Okay, so that's immune function. The other area where I think it's very clear that rapamycin has strong effects, we don't know yet in people whether it's going to work the same way, but it's certainly in mice, rapamycin has strong effects at rejuvenating the aged heart. And so there's work both from Simon Melov's lab at the Buck Institute and Peter Rabinovich, who's in my department at the University of Washington, showing that if you take old mice, again, 26-month-old mice in this case, give them 10 weeks of rapamycin, and then give them an echocardiogram before and after the rapamycin treatment, you can see significant improvements in cardiac function just from that 10 weeks of rapamycin treatment. And what Peter's lab has seen now is even 6 weeks is enough by echocardiogram to see improvements in cardiac function. One of the questions that they're very interested in asking is, how long does that effect persist after you stop the rapamycin treatment in mice? And I'm going to come back to this idea of cardiac function as a potential translational endpoint in a few minutes. So now I'll just show you two very preliminary experiments from my lab that I think are relevant for some of the things that we're thinking about in terms of potential translational value. So this is a very small pilot experiment. And the question that we were interested in asking is, how late is too late? So I told you that 20 months in mice, we can start treating them with rapamycin and get a significant increase in lifespan. But what we really don't know is whether there's a point after which starting treatment with rapamycin no longer improves health and improves survival. And so in this particular experiment, the mice were started at 28 months of age. So this 0 here is 28 months of age. As I said before, about 75% of the cohort has already died at this point. So this might be, I don't know, 75 years roughly in people if we were to try to do the extrapolation, 80 years maybe. And they either get a control diet, or this is now dietary delivered rapamycin in the diet. And so what you can see here is that the treated mice have about a 200% increase in life expectancy relative to the untreated mice. So, so this is, again, is a preliminary study. The cohort size is pretty small. Obviously, we need to have more animals to really know what the magnitude of the effect is going to be. But I think it's clear that starting very late in life, treatment with rapamycin can improve survival. This is particularly in C57 black 6 mice. Other studies have been done in other mouse strains that we can increase survival in very old mice. The other study that I want to show you, and now we've done this, we've replicated this now in a couple of different cohorts with a couple of different rapamycin delivery methods, is this question of how long do you have to treat with the drug to actually get some of these beneficial effects? And we started thinking that transient treatment might be effective based on the immune function studies that I talked about and the cardiac function studies that I talked about, where short-term rapamycin actually had positive benefits in those particular outcomes. So this is a survival study. We have health span measures as well that I can talk about if people are interested, but I think the survival data pretty much tells the story. So in this case, the mice are 20 months old when they come in. We treat them for 3 months with either control or rapamycin, and then we stop both of those and we just put them on a standard diet and put them on the shelf and see how long they live. And so that's what's shown here is the control group. This is now 23 months, so at the end of the 3-month period where they got rapamycin or placebo. The control group is here, and you can see they're dying off. This is about the kinetics we would expect of die-off in the NIA aging rodent colony, so the same strain background, same set of mice. And these are the mice that got 3 months of rapamycin. And so in this particular experiment, we don't know how big the effect is going to be yet. We're actually probably out here about now. So I can tell you it's at least going to be a 50% increase in life expectancy from 3 months of rapamycin treatment. Might be bigger. But it's certainly going to be statistically significant. It already is. And so again, that suggests that you can at least get some of the benefits of rapamycin for survival. And I haven't shown you the health span data, but also for health span, by just 3 months of treatment if you're a mouse. And again, thinking possibly translationally, that is potentially a more viable approach to translating this for human aging, in particular with a drug like rapamycin that does have some side effects. And probably those of you who know something about rapamycin are wondering about side effects. I'll be happy to talk about that in the discussion period. What I can tell you is the doses that we're using here are lower than what is used clinically in people. And these mice don't show the same kinds of side effects that is seen clinically in people. In particular, wound healing or immune suppression we don't see in the mice at these doses. But I think side effects are something that we need to pay attention to, and there are some interesting side effects in these mice. Okay, so how is this working? And this is really an area of ongoing interest in my lab and lots of other labs, and we have several projects around this idea of what is the mechanism. So it's sort of phenomenological that we can see these effects of rapamycin, but we really want to know at a molecular level in terms of pathways, how is rapamycin working? And I can't give you the answer. I can tell you some of the things that are probably involved. So rapamycin has many effects. It targets a protein, the target of rapamycin, that's involved in a lot of different cellular processes. And so it does a lot. And teasing out specifically of all the things that rapamycin is doing, which are most important for these effects on lifespan and health span is a challenge. So one of the things we know that rapamycin does that's a problem during aging is it decreases inflammation. So inflammation increases as we get age. It probably contributes to lots of age-related diseases or declines in function, and rapamycin is anti-inflammatory. So that's probably at least part of how rapamycin is acting. And one of the things that we know contributes to age-associated inflammation is this senescence-associated secretory profile. So senescent cells secrete pro-inflammatory factors into the systemic system. And rapamycin, Judy Campisi's lab at the Buck has shown that rapamycin actually attenuates that SASP. So that could be a big part of how rapamycin is impacting aging and age-related disease. Intracellularly, we know that rapamycin turns up this pathway called autophagy that's involved in breaking down damaged macromolecules, including entire mitochondria. And there's lots of evidence in the literature that that's probably involved. Rapamycin also affects protein synthesis and mRNA translation. And one of the areas my lab is very interested in is the effects of rapamycin on mitochondrial function. So we published a paper about a year ago showing that rapamycin could impact mitochondrial disease. So this is a mouse model of Lee syndrome. It's a childhood mitochondrial disorder. It's actually the most common mitochondrial disorder in people. And what we saw was that rapamycin treatment in these mice was able to more than double life expectancy in this mouse model of severe mitochondrial disease. I'm not going to try to argue that aging is mitochondrial disease or that mitochondrial disease is accelerated aging. I do think that it's clear that mitochondrial dysfunction increases as we get older and as other animals get older, and that to some extent aging could be an acquired mitochondrial disorder. And so we think that the effects of rapamycin on mitochondrial function in this disease model may also contribute to the effects of rapamycin on age-related disease. And then we're also interested, this is a fairly new project in the lab, our effects of rapamycin on the microbiome. And I'll just show you a tiny bit of data on that. And so we first actually started thinking about this by looking at poop, believe it or not. So this is, as you might be able to tell, mouse feces. And one of the things we noticed was that in our rapamycin-treated mice, they had small poop. Actually, I didn't notice this, my postdoc did. And I started laughing when he showed this in lab meeting. So we thought for a while, what does this mean? What could possibly account for small poop in a mouse? And there's lots of trivial explanations. Maybe the mice are dehydrated. But one of the ideas that occurred to us is maybe there are changes in the microbiome in rapamycin-treated mice, and nobody had ever looked at this in the context of rapamycin before. One of the interesting things we noticed as well was that this phenotype appeared to persist after we stopped the treatment. So when the mice came off of rapamycin, they still had small poop. So we looked at the microbiome. This is a collaboration with Catherine Hagen at Missouri. And this was initially done by just sequencing DNA in the feces. And then we followed up by actually looking at histopathology from the small intestine. And in both cases, we saw, as you might expect, anything's going to change the microbiome, right? We saw lots of changes. The one that was most striking to us was this dramatic increase in segmented filamentous bacteria, or SFB. And you can actually see them here as these long filaments in the intestine. And then in DNA analysis and RT-PCR, we were able to detect increases in SFB in at least 4 different cohorts, both by injection of rapamycin or by dietary treatment with rapamycin. So we are, this is rock solid. We're absolutely sure that at least in C57 black 6 mice from the NIA aging rodent colony, rapamycin always leads to this dramatic increase in SFB. Why are SFB important? Well, there's a growing interest in the microbiome community of the role of SFB both in metabolic disease and in inflammation, and we know that rapamycin can impact both of those things. So we're wondering if these effects of rapamycin on the microbiome may contribute to some of its effects on inflammation and metabolism, and so we're actively studying that. And I do think in a general sense this also raises an issue that lots of people are starting to recognize, but that I think we still don't understand, which is what is this interaction between drugs that are given to people or mice and effects on the microbiome. I think we've generally thought about the effects of drugs on cellular function, and we haven't really appreciated that lots of drugs probably are going to impact the microbiome as well. So this is something that we're looking at, whether this might be involved in rapamycin's effects on aging. So now I'm going to switch gears and just for the last 5 minutes or so talk about how we're trying to address what I really view as the biggest challenge, at least from a translational perspective in my field. And so I've already told you, we've done pretty well at figuring out genes that are involved in aging, identifying some drugs that seem to impact aging, and showing that that's true in laboratory systems. How do we actually start to translate this in a meaningful way into people? And there are, lots of other people in the field have thought a lot about this, obviously, and there's lots of people who are interested in this idea. I think the most obvious approach is clinical trials, right? So you might, let's say rapamycin. We've got a drug that we think affects aging. We want to do a clinical trial to see whether it affects aging in people. There are some challenges with that. One of them is that to really do that, to really assess healthy aging in people, we're probably talking a 2-decade clinical trial. So that's challenging. The other challenge is that FDA does not recognize aging as an indication or a disease, and so you actually can't do a clinical trial for healthy aging now. People are talking to FDA and working with them. Nir Barzilai at Einstein, you may have seen this in the New York Times, has proposed a clinical trial of metformin. They're calling it the TAME trial, Targeting Aging with Metformin. And they're trying to work with FDA to actually assess whether metformin improves healthy aging in people. And I'm happy to talk more about that if you're interested in a few minutes. So that's one approach. My real concern with that approach, though, is the strategy that most people are taking is to say, we think that a drug affects aging, therefore it probably affects age-related disease, therefore let's try it in a clinical trial for an age-related disease, Alzheimer's disease, for example. My worry with that approach is that once the disease has reached clinical diagnosis, it's not clear that it's normative aging anymore at that point. And Alzheimer's disease in particular, I think is easy to understand in that way. So an intervention that is, in fact, hitting aging at a molecular level won't necessarily be effective once the pathology of the disease has progressed to clinical diagnosis. So I worry that that approach runs the high risk of failure. And as we all know, failure is probably the most common outcome of clinical trials. So you really want to think things through before you go down that road. So the approach that we're taking is to ask, what is there some intermediate that has value in and of itself that can also then inform us about translation to people? And the approach that we're taking is pet dogs. So this is actually my dog. His name is Dobby. And there are lots of reasons why we think that testing rapamycin in particular in pet dogs now makes a lot of sense. And if you're interested, this is the website. It's very easy to remember, Dog Aging Project. So why does this make sense? I'm not going to read the list to you, but I think there are lots of reasons why this makes sense. And I just want to mention a couple. that I think are particularly relevant. So as you all probably, how many of you have dogs? Just out of curiosity, okay. So those of you who have dogs, you probably have recognized that our dogs get old, unfortunately, very quickly, right? And they get many of the same age-related diseases that we do. So they age a lot like us, but they do it much more quickly, particularly if you have a big dog like I do. Big dogs age faster than small dogs. So one of the really powerful approaches to this particular strategy is that we can know the answer in a relatively short period of time. So I told you that rapamycin, starting at 20 months in a mouse, significantly extends lifespan. If the same thing is true in pet dogs, then if we start in a mid to large sized dog at about 9 years old, we're going to know the answer in 3 to 5 years if we're appropriately powered. Okay, because dogs age fast enough that this is actually a reasonable time frame to answer this question. They've got a lot of genetic diversity, but a big one is that they share our environment, and probably for some of you, they share your bed, right? I mean, dogs share our environment in a way that really no other animal can match. And then one of the things that I've really appreciated about this is that it also engages the public in citizen science. So those of us who love dogs really love our dogs. And I think this is a scientific endeavor that the average person can understand and be enthusiastic about it. In fact, I've actually had a couple of teachers contact me and say, hey, I want to use this as an example of science in my class, because people really like this idea that we can improve the healthy longevity of our pets. So how are we doing this? So we actually have a rapamycin intervention trial that's about to get started. The idea is to start with large breed dogs and a low dose rapamycin to start them in middle age. The initial study will be a short-term study. The longer-term study will be a 3 to 5 year study where we can actually assess not just survival, but age-related health parameters, so cardiac function, immune function, cancer rates, mobility, cognitive function. All of those can be assessed in dogs. And we can actually assess whether rapamycin is delaying a wide variety of age-related declines in function or is primarily affecting survival. And our prediction is that the treated animals will be healthier and live longer. And so we've had more than 1,200 dogs enrolled, most of them in the US, but we do have some international dogs as well. The phase 1 trial is actually starting in about a week. The first dogs will be coming into the clinic and will be randomized into placebo or treatment group. It's going to be a small study. It's mostly a dosing study, and then cardiac function we're also looking at as an endpoint where we hope to see improvement. About 40 dogs. This is my other dog. Her name is Chloe. This will be done at a VCA specialty clinic in Linwood. So the dogs will come in. They'll get an exam, some blood work, and a baseline echocardiography. Assuming no pre-existing health concerns are noted, they'll be randomized into placebo or treatment group. 2 weeks later, they'll come in for another blood draw. And then after 10 weeks, they'll come in and they'll get another echocardiography. And we'll hope that we see improvements in cardiac function at the end of the 10 weeks. And then if we're successful at raising funds, if any of you have $15 million, we're happy to have a good cause for you if you think this is a good idea. But seriously, if you like the idea, we're on Facebook, we're on Twitter. Please get out there on social media and talk to us. We hope to expand this into a national study in about 3 to 5 years. And one of the reasons I'm really so excited about this is I really think that within 4 years, we could actually have an intervention that can significantly improve healthy aging in our pets. And to me, that's really cool. So this is where I'm putting a majority of my effort right now. So thanks for your attention. I just want to mention Daniel Promislow in the pathology department at UW is my colleague on the dog aging study. And I'll be happy to answer any questions. Thanks.